Hello, this is your instructor Kwame Shen. So we will continue from the first part of Introduction to Microbiology and Microscopy. So this is part two. With part two, we're looking at a brief history of microbiology in terms of the various developments that we made incrementally over the years, actually over centuries. We'll look at the objectives. For objectives, there are some key scientists, and I want to emphasize that every scientist who contributed significantly to the growth of the field of microbiology and to the betterment of the human species played a keen role. However, there are some significant ones that we'll highlight in the course of our lecture for this particular topic. We'll look at Hooke, Van Leeuwenhoek, spontaneous generation and biogenesis, the whole debate and how it was eventually resolved, the work of Pasteur's, Koch's, Jenner, Ehrlich, and Fleming, in addition to some other microbiologists. We'll also briefly touch on microscopy because of the work of Hooke and Van Leeuwenhoek, and the fact that without microscopy we couldn't have made all this development that we've made so far. Robert Hooke in 1665 observed that when he looked using his very today we'll look at it as very archaic, but it was a very ingenious on his part to create this apparatus which helped him magnify and be able to see closer than scientists had been able to do previously. So he observed Koch cells, and they were not called cells at the time. So slices of Koch that he put under a special instrument that helped magnify the contents of the Koch. And what he noticed was that they were made up of these tiny spaces. There's several of them. And as far as he was concerned, they looked very similar to the cells that he lived in as a monk. Cook was a monk. And so the monastery cells became the name of what today we call cells. Eventually, it was realized that all living things were made up of these tiny components, and so that led us to the cell theory that all living things are composed of one or more cells. Cell theory can be rendered in various forms. So the other two are also the same. They're saying the same thing differently. So cells are the basic units of structure of life and function as well. And cells are needed. That without them, new cells would not come into existence. So pre-existing cells are needed for new cells to come into existence. So after Cook's discovery, Anton van Leeuwenhoek also designed a magnifying instrument like a microscope. And using his microscope, he observed several microorganisms, live ones. He collected water from pools, seminal fluid, and then he saw very tiny organisms that were living. He called these organisms animalcules. He called them animalcules. And these are some of the drawings that he made of his observation. So he had pepper water, hay water, dunghill water, and he saw all these microorganisms in there. He did that for seminal fluid and other water bodies that he collected and observed, and he saw that there was a whole world out there that we had no idea about. So that was basically the birth of microscopy, because once we noticed that there is a whole living world out there that we previously had no knowledge of, it led to the birth of microscopy. What microscopes do is that they give us the ability to see beyond what naturally would have been able to see. One of the important characteristics of a microscope is its ability to resolve between two objects or 2 points, so that you can tell them apart. So for instance, if you look at this, the picture on the slide, at the far right, it's so blurry because the resolution is very little. You cannot tell 2 points apart clearly. It gets better because these are 20 pixels, it gets better than the 50, and way better than the 70. So the difference between 2 points is more distinct. You can tell them way better when you look at the far left image than you look at the far right image. Microscopes employ refraction. It depends on the type of microscope, but some would also use reflections and diffractions from the object or specimen of interest as well. And the whole point is to make the objects appear bigger and the images clearer, so that we can actually see details that otherwise wouldn't have been able to see. Microscopes are limited. For instance, if you take light microscopes, they are limited by the wavelength of white light. They can't resolve structures beyond a certain size. And so if we look at the wavelength of light, as you go towards ultraviolet, towards the violet, the wavelength decreases. As you go towards red, the wavelength increases. So the type of light that a microscope uses may help you to actually see better than others. We generally will use light microscopes for a microbiology class as ours. Light microscopes may come in the form of one lens or a compound lens microscope with varying magnifying powers. They would have an ocular lens, and then they would also have objective lenses. There will be a set of prisms here. There will be a source of light and a condenser here. And the whole point is to refract or bend light, that as it goes through the object and comes to the ocular, it is magnified, and we can see more details than we otherwise would have if we didn't have the help of the microscope. There are other light microscopes that employ different techniques, like dark field microscopy. So for light microscopy, the ones that we generally would have used in the lab, it would use bright illumination, so it just white light, and we don't really manipulate it much. As it goes through the lenses, they're diffracted and we're able to see. But to be able to see that, there is an issue of contrast. So on the light microscopy, usually we would stain the objects that we'll observe. First of all, we'll fix them on a slide using heat, and then we'll stain them with dyes to create more contrast so we can tell them apart. The process kills these organisms that we are observing. And so although the structures are generally maintained, because the organism is killed, it affects what we're able to observe. Dark field microscopy is used to observe live microorganisms that we cannot observe under light microscope, the visible light microscope. So it employs a strategy where you have a dark object, an object that is used to block the light rays, and then you only have reflected light from the object that enters the objective lens, and so you're able to see the object lying in a very dark background. That is how Treponema pallidum is observed under the microscope using dark field microscopy. We could also use a phase contrast microscopy, where we are able to see more details in terms of the internal structures, and it all comes down to how the lenses are positioned and all that. Then there are other microscopes that would use sound to actually create images, and so they're able to resolve better than light. There are others that will use electrons. Electrons also, like sound and light, are waves, and so once they go through, these waves are beamed through, of electrons are beamed through the objects, they are refracted, and then they are magnified. We have basically two main types of electron microscopes. We have the scanning microscope and then the transmission microscope. The scanning electron microscope has a resolving power of about 10 nanometer. The transmission electron microscope has a higher resolving power of 0.2 nanometer. So the objects are projected onto a screen. That's the microscope, the electron microscope. The object is projected to a screen, and then it is observed that way. So from these comparisons, based on the property that is employed, if you use a light microscope, you can see beyond 100 micrometer, which is the limit for most of our eyes, but you can see all the way to 1 micrometer. Then you have electron microscope that can see beyond 100 nanometer. Okay. So for scanning tunnel microscope, atomic force microscope, these are more powerful than electron microscopes. So you can see amino acids and the likes with these and proteins. For the electron microscopes, you can see internal structures of cells with that, viruses as well. With our unaided eyes, we can see up to 100 micrometer. Compound light microscope, we can see from lower than 100 millimeter all the way to around 100 nanometer. So once we discovered that microorganisms do exist, the question then was, where did they come from? Where did these microorganisms come from? There were two competing theories at the time. One was that they sprung up suddenly. They sprung up spontaneously because of a vital force that exists in the air. And so that was one hypothesis. The other one was that that living organisms could only arise from pre-existing living cells. And so they came from other living organisms. Science always operates like that. There are sometimes several competing explanations for an observation. And so experiments are then conducted to determine which one is really the correct one. For spontaneous generation, it made sense that if you put a piece of bread somewhere, you might find mice coming out of it, or you have meat or fish there. After a while, you might see maggots coming out of it. So these animals, in terms of the mice, or these microorganisms, in terms of maggots and the likes, people thought they just arose. They arose spontaneously. Until 1668, when Francesco Redi filled 6 jars with decaying meat. So this guy is going to conduct an experiment to actually determine whether microorganisms arise spontaneously or by arising from pre-existing cells, which we call biogenesis. He had 3 sealed jars with meat in them, and 3 open jars with meat in them. The result was that the sealed jars had no maggots developing in them, so they stayed okay. I'm assuming it was for a short period of time, because over a longer period of time, the meat would have gone bad because the bacteria in the anaerobic bacteria would have acted on it. And then the open jars became infested with maggots. And so the open ones, we know now that the maggots came from flies that lay their eggs on the meat. However, at the time, they didn't have this knowledge. So this particular experiment was a big blow to the hypothesis or the theory of spontaneous generation. But the proponents of spontaneous generation said the issue with this design is that it blocks off air from getting into this flasks. If air is blocked from getting into the flasks and vital force is in the air, then of course you're preventing vital force from acting on the meat for the microorganisms to arise spontaneously. And that is how science often operates. You see something, you come up with a result, and people challenge it, and you have to come up with a better explanation. So Redi did another experiment, and this time around, what he did was he introduced a third set of jar, which he covered with gauze. And so he covered that with gauze. So obviously now the flasks will have, the meat will have air circulating in it freely from the outside. Vital force will not be denied access to it. And what happened was that there were no maggots in the ones that he covered with the gauze. And the first 2 also was the same. The open jars became infested. The closed ones were not infested with maggots. The question then is, where did the maggots come from? And we know they came from these flies. But that wasn't the end of the debate. In 1745, John Needham came up with a very, very interesting explanation for that observation. And what he did was that he boiled chicken broth and covered it, and over time they were infested with microbes. They went bad, and so the theory of spontaneous generation again was backed up by his research. Until Lazzaro Spallanzani also boiled the same chicken broth, and then what he did was that he kept some of these jars opened, and the others he covered them. However, there were no microbial growth in the ones that he covered. So the explanation that Lazzaro Spallanzani gave in terms of the results that John Needham got was that Needham didn't close the flasks that he used in his experiment. After he boiled it, he didn't close it fast enough or quick enough, and that microorganisms were able to access it before he closed it. In 1853, Rudolf Virchow challenged this whole idea of spontaneous generation by just his hypothesis. And his hypothesis was that living cells arise only from pre-existing living cells, the theory of biogenesis. He had no proof, however. But it was around the time where Louis Pasteur had demonstrated that microorganisms were present in the air. Pasteur came up with an elaborate design where he heated the necks of very long flasks. He put broth in it, beef broth, heated the necks and bent the neck like that. And then after he did that, he boiled it and left it sitting. And these broth never went bad. And the reason is that these bends that he had here prevented the microorganism from gaining access to the broth. So there is no covering or any sort, and he ended up disproving the theory of spontaneous generation. This led to the Golden Age of Microbiology from 1857 to around 1914, which is described as the Golden Age of Microbiology. There were several discoveries around the time, and science in terms of microbiology made a lot of progress at the time. Louis Pasteur, who is a French chemist, is described as the father of microbiology. In addition to disproving the theory of spontaneous generation, he identified organisms that were causing infections in silkworm, which was used in the fabric industry. And in addition to that, he played a role in identifying the causative agent of cholera, chicken cholera, vaccines as well for rabies and anthrax. He played a role in that as well. One of the important things that Pasteur, as his name is, also demonstrated was that microorganisms were responsible for fermentation. So he did that when he was working in the wine industry, and then he also demonstrated that when certain food or beverages such as wine are heated just enough, they could kill all the microorganisms that could cause it to go bad without destroying the taste of the food. And we still use these processes today. That's the process of pasteurization, where we apply very high heat over a short period of time to preserve milk and the likes. This led to the germ theory. The germ theory being that microorganisms may cause diseases. In addition to these developments during the Golden Age, you had Ignaz Semmelweis, who, after having this knowledge that microorganisms may be the cause of diseases, may cause diseases, and the understanding from Louis Pasteur's work that microorganisms exist in the air and all that, he advocated that physicians at the time should wash their hands when they are performing obstetrical surgeries. There was a pushback, but eventually got his way, and it was shown that it drastically reduced infection. So basically, this guy set out the first rules of epidemiology and disease control. Using a similar knowledge, Joseph Lister built on that, and then he used disinfectants such as phenol to sterilize surgery equipment and clean wounds, and it was also shown to be effective. Now, up to that time, there hadn't been a direct link between a bacteria and a disease. Robert Koch was the first person to show that anthrax, which is a bacteria, anthrax, which is a disease, is caused by the anthrax bacteria. And that led to what we call the Koch's postulate. For the Koch's postulate, it stipulates that, first of all, you should, if a disease, an organism is, an animal is killed by a disease, you should be able to isolate the microorganisms that you hypothesize or you believe may be responsible for the organism's death, isolate it from the dead organism, be able to culture it and observe it under the microscope, identify it, and then you introduce these microorganisms that you've isolated into a living living healthy animal. If the disease is reproduced in the healthy animal and it dies, you isolate the microorganism from this healthy animal which just died from the reproduced disease, and identify it again under the microscope by culturing it and observing it under the microscope. If you're able to do this, then it means that you have actually proved that there is a link between that particular microorganism and the disease. So that's the Koch's postulate, which is still used today. There are limitations to this postulate, however. and we'll talk about it briefly later on. And 70 years before Koch was able to come up with a postulate, the first vaccination as recorded in the Western world was done. It is believed that this had been going on in China in the 1500s before the Western world started vaccinating people. And it was mainly based on the understanding at the time that Edward Jenner had that milkmaids who every now and then will suffer from cowpox virus were not affected by smallpox outbreaks. And so he took blisters from cowpox blisters and used it to inoculate an 8-year-old boy. It sounds very unethical, but that's how it started. And this boy had a mild form of the disease at the time, recovered from it, and never suffered from smallpox. So that was the beginning of vaccination. Vaca, that is for cow, from the cowpox virus that was used at the time. So, and the boy was able to develop immunity, which we'll talk about again. So look at what has transpired. We discovered a whole world of microorganisms, living organisms we didn't know. We're able to establish that these microorganisms come from existing microorganisms. We're able to show that there is a link between these microorganisms and certain diseases. And so we have vaccination. And then that led to the treatment. Once we know there's a link and people were able to protect themselves from getting certain infections like disinfecting equipment and the likes, it led to the pursuit of treatment. So we're looking then for ways that we can actually destroy these microbes that cause diseases. And so we have antibiotics, which are natural chemicals that are produced by bacteria or fungi and are able to kill bacteria. And then we have synthetic drugs. So these are artificially made in the laboratory. And the aim is also to destroy disease-causing microorganisms. So the first synthetic drugs, as we know, had been in existence for several years, actually centuries. The quinine had been used for so many years in Asia to treat malaria. But in terms of bacteria, we actually didn't have any synthetic drugs until Paul Ehrlich speculated that there could be a magic bullet. And what he means by that was that there could be a chemical that could be synthesized which can destroy the pathogen without destroying the individual. So that's the goal of it being magic. Destroys the pathogen or disease-causing organism without destroying or causing harm to the host. So in 1910, he developed a synthetic arsenic drug called Salvarsan to treat syphilis. Salvarsan was called Salvarsan because it was seen as a salvation drug for syphilis. And there was a lot of pushback at the time. There was a lot of side effects, but he was able to produce a less toxic form of it that was used for a while. And this knowledge resulted in the development of sulfur drugs called sulfonamides, which were very important during the Second World War in treating thousands of soldiers. Then later on, in the 1940s, penicillin was then used in a mass scale. And for that to have happened, in 1929, you had Alexander Fleming accidentally discovering antibiotics. Fleming had incubated Staphylococcus aureus, and he noticed that just when he was about to throw his plate away, he noticed that there was a portion around the mold, a mold that had grown on there. There was a portion around the mold that the bacteria didn't grow. And so it was discovered that this bacteria produced an antibiotic called penicillin, which was effective in killing several bacteria. And that was the beginning of antibiotics. We were able to produce it in mass quantities in the 40s, which was the second golden age of microbiology. And subsequently, we continue to develop in terms of our knowledge of microorganisms. We eventually entered the third stage of third golden phase of microbiology. Then 1990s to the 2010s and beyond, we still continue to discover new things in terms of microbiology using genomics, the genes, studying the genes, and being able to understand how best we can protect ourselves and enhance ourselves as a species. So this brings us to the end of our second part of the topic, Introduction to Microbiology and Microscopy.